Hello, hello. Hi. Hi.
I'm not getting anything. Oh, there we go. As soon as I say you're good.
Can you hear me though?
I haven't. Yep, you got sound.
Sweet. Hey y'all, what's up? Look at this light. Oh my god, it looks so beautiful.
Wow.
This light is everything. It's my Instagram light. I conduct makeup tutorials with this. This is the pipette tips, or not the pipette tips. These are inoculation loops, as you can see. Yep. Really nice for my complexion. What's up, Josh? What's up, Brandon B? Julia Miller. What's up? Like my shirt? Okay, let's see. All right. What's up, Bill? What's up? Okay. Looks like a bunch of people are coming in right now. I'm gonna kind of wait a few minutes. If you guys haven't started on the experiments, don't worry. I heard Jill did a great job with teaching you guys pipetting and scale balancing. That's pretty cool. Oh shit, that's 101. Never mind. Josiah taught how to use Snapgene and things like that, which is pretty cool. Yeah. Oh, Mr. Matt Barnes. Burp, burp. Trying to figure out what's like the best. Maybe like a parrot on the shoulder? Or like... I'm actually grabbing with my mouth. It's probably not sanitary. Okay. All right. No, David. It's just me. Okay, we'll get started. So, my name is Esther. I am your other instructor for this class. There are many instructors for this class. As of now, there's 3. And you've met all of them. It's Josiah, Jill, and me. So people that haven't taken the 101 class, I am the technical instructor. So I'm here to guide you on your way to genetic engineering. Yeah. Smooth jazz. So today we'll be talking about incubation and mini prep plasmids. So this is kind of a work in progress where we're doing this on a moving camera, so it might be shaky. Just let me know if anything needs to be made clear, or if you can't see any of the vials, just let me know. Jill will give me a shout out and I'll be sure to correct it and show you again. So this is kind of a quick and dirty tutorial. I just want to preface that these YouTube tutorials are not necessarily the whole class. Now, if you purchase the class, you will get all the material sent to you. You'll get access to the Google Classroom, and everything is accessible through there. The YouTube channel is just a supplementary piece of material that you can use as a reference guide, or just a little something extra so you can kind of learn from it visually. So without ado, I will show you how to do a mini prep. Cool. First of all, we got these nice little gloves that we've provided for you. And put them on real quick. Let's talk about incubating. So a portion, let's see, hold on, 1 second. Sorry, we had a last minute like changeover of the computer, so yeah. All right. So in week 2, we gave you a document called DIY Incubator and Growing Bacterial Culture. So if you access that, it'll show you how to grow up things on a heat mat. So we provided you with a heat mat that you can incubate and incubate your bacteria and whatever you'd like. So generally, these are all in Celsius, thank the Lord. So bacteria generally grow best in 37 degrees Celsius, and especially the strain that we gave you grows very well in 37 degrees. With other different organisms, they all vary in temperature, what keeps them happy. So whatever your organism is made happy, that's fine. Just do your research, do your homework, see what they grow best in. Now, we also mentioned the use of antibiotics. Now, from the 101, we learned that antibiotics are a good way to weed out the competition, to obtain something and select for a specific thing. So generally, when we engineer bacteria or yeast or any organism, we want to know that it's like a pure culture. We want to know that it's the correct thing that we're looking for. So we generally add a marker in the bacterial plasmid that we synthesize, and that indicates that if it's antibiotic resistant. So that's just an easy way to show that this is the correct colony. This is what you want in the end when you genetically modify something. So when it goes into a broth or like a LB agar or like a plate, like you don't really know what's growing unless you put antibiotic selection. So you had to do a few calculations for kanamycin and ampicillin, and you can do 1x concentration of that. And the document clearly shows you how to do some of the calculations. If you have trouble with maths, like I do, just give me a shout. I'm more than happy to accommodate you. Esther at the-odin.com, or find me on Facebook or Instagram. I am readily available all the time. As some of my students can tell you, I sometimes drink a beer on the weekends and answer some of your questions that you have. Yeah, I'm fucking dedicated. So, let's see. I did start a starter earlier today. It didn't make the 24-hour period. Let me go see if it's like grown at all. Oh, like the away. Yeah, so I started a pea dawn culture, and earlier today at maybe like 5 hours ago, and it obviously hasn't grown very much. It's still looking pretty clear, unfortunately. So I'm gonna throw this away. But generally, you start the culture overnight, and generally within 24 hours, you want to take the culture out. So what you do is you take one of your nice little sticks, and you inoculate a broth culture, and you grow it up overnight for 24 hours. Now, you can tell that if it's working or not by how clear it is or how opaque it is. So if you take the culture and you generally do like a negative control, so you'll do 2 tubes. You'll do 1 tube without any bacteria and the other tube with bacteria. And then you grow it overnight, and then with the antibiotic, of course, and then you grow it overnight, and then you figure out, okay, did it grow or did it not grow? And if it did, how much culture do I think I have? So if it's like very, very cloudy, as the picture shows in the document, in the Google document, that means it grew. And so if it grows, it's very like flocculent, it's very like opaque, you can't really see through it, then that means you did a great job and your organism did a great job replicating itself overnight. And so then you can start your experiment. This is something that I grew a while back. You see it's kind of like really gross and boogery. I don't know if you can say it. But it's kind of like this like really weird culture. No, I guess you can't really see, it's kind of like whitewashed, but there is a culture in there, and it's very, very brown. It's very thick. I kind of defrosted it. So this is what we're going to be working with today. So you kind of mix it up. It's kind of chunky, and then they're usually in. So once you grow that up, then you're going to spin it down in microcentrifuge tubes that we have provided for you. Yay. Let's see. So I'm going to show you a few things. Here is our mini prep kit that we got, that you must have received from us. And note that some people's buffers have exploded during the transit over to you. So if they did explode, make sure you contact me or Josiah or Peter, our lab manager, Peter at the-odin.com, if you need replacements. I know that they spilled on the way and we didn't account for the shipping methods because it's our first time shipping these kinds of buffers. So just let us know, we'll replace them right away for you. So what you should have gotten in your kit in order is the A1, buffer A1, buffer A2, buffer N1, column buffer, Elution buffer, and DNA wash buffer. Oh my god, so many buffers. God damn. And we also got these really nice, very unique wash columns. Fun. Great. Okay, so after you grew them up for 24 hours, then you take these 1.5 empty little tubes and you fill them with about 1 microliter of your grown-up little plasmid. Go ahead and do that. Hold on. Hey Jill, can you help me film? Okay, well, this is our screen, and this is our camera. So if you wanted to, just go over. Okay, so we have a few tubes here. Now the important thing to centrifuge balance is that it needs to be balanced. So whatever goes on one side needs to go on the other, or you ruin your centrifuge, which has happened a lot. So I'm gonna take the the 1,000 pipette tip. We're going to change it to 1 milliliter or 1,000 microliters. 1,000. Put a little nice tip on it. And we're going to take this goopy gRNA that I spun down and pipette. Close that up. And then conveniently throw it away. Okay. Pamela Lewis, you do 1 microliter of the culture that you have cultivated overnight. Yes. All right. So whatever is done to one tube must be done to the other tube. That's what a balance is. So I have some handy dandy DI water. You can use sink water. You can use any type of water. You don't even have to use water. But just make sure that when you do this, the same volume should be on the other one. Because why? We need to balance the centrifuge. So that's about the same volume. No, it's not. It's the water. It's okay. Aha. So it's the same volume, as you can see, of water and our plasmid, our bacteria. And you're going to take your really cool new centrifuge. Ta-da! Ta-da! You guys are all proud owners of a beautiful 10,000 RPM centrifuge. And you put that in one end, and the other one, the other counterbalance, you're going to put directly on the other side. And you got to make sure that it's balanced, as you can kind of see over here, because what happens is if you misbalance it, or if you put the wrong amount of liquid in one of them, then it can grind away at the centrifuge, and it'll just not last very long. So just make sure that every centrifuge that you ever operate is fully balanced. So, fully balanced, different opposite sides, ready to go. So now this only does one type of speed. I understand that in the protocol it's like spin at 9,000 RPM or 13,000 RPM or 16,000 RPM. It's just like a bunch of RPMs. For the purposes of mini preps, I don't find that RPMs necessarily matter that much, as long as it breaks a certain threshold so the cells can lyse. That's fine. So we've experimented with the 10,000 and it's worked very well. Anything under this, I haven't really gotten such good results. So it's fine if you don't follow along with the, I believe it was the plasmid isolation from BioLand. So it's okay for the purposes of this lab and future mini preps. You're completely fine with using 10,000 RPM. So let's let it rip. We're gonna put it down and we're going to spin it. Buzzwordy 09, this many G's. I'm not sure how many G's translate into RPMs, but this is a 10,000 RPMs. And it's sufficient for all of your mini prep experiments. Okay. So in the protocol, it does say 5 minutes, but I'm not going to bore you with 5 minutes of spinning. So I'm just going to hypothetically just make sure that a pellet forms on the bottom of the tube. So this is obviously not pelleted, but the pellet will look very, it'll look very pellety. It'll just it'll just be a small little like lump on the bottom, and the liquid should be completely clear. So that's when you know it's been pelleted. So a good measure is centrifuge it on here for 5 straight minutes, and then once you take it out, there should be a very nice pellet on the bottom. All right.
We got a couple questions.
Sure. So, I think Josiah said 3,000 Gs. 3,000 Gs won't cut it, Julie Miller. That's why all of our centrifuges that we give to the 102 class is all 10K. T. Williams, you couldn't see that. Are those the Eppendorfs or the little tube thingies you guys sent? So these are the 1.5 microcentrifuge tubes. And the Eppendorfs are a brand of pipette, and we do not use that brand of pipette. And whatever we sent you, or since you took the 101 class, you can just reuse those pipettes that we've sent you. Let's see. All right, yeah, so Pamela Lewis, like the book says 5,000 Gs. Like I said, it doesn't really matter. We ourselves at the ODIN have tested these. So at 10,000 RPM, everything should lyse. Everything should be good. Anything, if you go any lower, unfortunately your cells don't lyse quite as well, and so you won't be able to separate the DNA from the plasmids or the rest of the junk that you don't want. So RPMs, Gs, they should be all good for our experiment. All right, cool. Zayner, for the purposes of this experiment, we're just going to use a little bit of water, which I hope it's okay with you. Because, again, I don't want to spend 5 minutes watching something spin. Okay, so then next you're going to see the pellet, and then you're going to add so you're going to see a pellet, hypothetically, on the bottom here, and you're going to take your 1 ml pipette and you're going to pipette out all of the liquid. Okay, and you can discard that whenever you please. So what you're left over was with a small hypothetical pellet in your 1.5 milliliter tube. And then after that, you're going to play with your buffers. So you're going to adjust your pipette to 250 microliters of buffer 1A. Now you can follow along with me from the Google Classroom document, or you can watch this like really quick and dirty, easy way of doing it through this YouTube channel. But yeah, so you know, whatever enforces your learning, you could totally do. So you could follow along, or you can watch this YouTube tutorial and then go back. So I'm reading literally right off of our document, and it has 250 microliters of buffer A1. Big crank down. Okay. So again, it's 250 microliters, yay, on our 1000 microliter pipette. Yeah, for your nuance, you can just follow along with the protocol. Again, this is just a supplementary material for people that can like to visually see. So you have your pipette ready, and you're going to take one of these really nice columns, these really fancy columns. They are much more expensive than the regular microcentrifuge tubes, so use them very, very well and very, very smart. Okay, so you're going to take the pellet, and you're going to take 250 microliters of the A1 buffer, pipette it up, and then drop it in, and you're going to just slightly pipette up and down to mix up the pellet, to mix up the buffer. This is just a very gentle way of treating your cells with respect. So much respect. Yeah. So once the pellet is broken up and mixed, you can give a little shake. Then you're going to take this pipette tip and discard it. So now this is A2. So remember, when you're dealing with buffers, you must close everything like I didn't do, but make sure everything's closed, everything is sterile, everything's closed. And so we take the A2 buffer, and again, we get a brand new tip from the box. Open it up, and again, it's 250 microliters. So we're gonna pipette that up real quick and close it right away because we don't want to get any of our buffers contaminated. Yeah, you see that one-handed. Booyah. And you're going to just add it to our solution. Right? Throw this away. And then what you're going to do at this point, you're just going to invert it. Now, at this point, the DNA and all the junk, the cell junk, is going to separate. And it's going to create this cute little bugger. It looks like a loogie. It's kind of gross. But you invert it very carefully because the DNA and the cell debris, such as the proteins and also the cell wall, the lipid layer, will all separate at this point. So you kind of invert it very, very gently. And once that is happening, you can't really see here, or because number one, this is fake. Number two, like you can't really see here, but you'll see like this goo forming and this separation forming. So then you're going to set it on the bench and leave it for 2 minutes. So you leave it for 2 minutes and you come back in 2 minutes. So then the next step after that would be to pipette 350 microliters of N1 buffer. So I'm going to change that real quick. Oh, I know, a 4-year nuance. I feel so dirty too, not being able to do this in a hood, but honestly, science can be done on a lab, on like a kitchen. It's just crazy. Like, we've had no contamination errors or issues here at the ODIN, so why can't you? All right, so this is set at 350 microliters, and we're going to take the third buffer, the N1 buffer, and we're going to add 350 microliters to our boogery mixture. So at this point, you should have about like 800 microliters in your tube, which will all fit. Now, if you did this incorrectly, then you have way more and it should be overflowing. So, yeah, and then it precipitates out more. So then you close the tube and you have 3 things in here, A1, A2, N1, and you shake it very vigorously. This will cause even more separation from the cell junk from your DNA. All right. Even here, you can see a bunch of little floaties. It's much more opaque than like water, right? So then you're going to leave that there, and you're going to place the tube in the centrifuge for 10 minutes. Now again, you can use your water balance, but just make sure that everything is balanced. So again, this is just water that I'm going to be taking, and I'm going to balance like the 800 microliters I have here. Just visually, I can just see and add water. Just to make sure that our centrifuge doesn't get ruined. Yay. Alright, and we're going to centrifuge it again. Again, what? Keep it balanced. So we're gonna centrifuge it. Now this portion takes 10 minutes. So I'm not going to spin it for 10 minutes, but it's kind of cool. You can see a small little pellet that formed. Now, I don't know if you guys have ever taken like an organic chemistry course or anything. Probably not. But there's something called the baby. Now the baby is something that you don't want to throw away. There's going to be lots of steps within the mini prep, and it's very important you don't throw the baby away. Now the baby in this case is all of our DNA, and our DNA at this current state is in the liquid. It's not the pellet. The pellet is where all the cell debris went. The pellet has the lipids, the proteins, all the junk you don't want. And since this is a mini prep for DNA, we want to keep the liquid. We want to keep the baby. So just keep that in mind where the DNA is after every single step. And that'll help you visualize where everything's going. So a really nice pellet has formed even within the seconds that I've been spinning it down, which is nice. So we're gonna keep this here. So then you're going to take out all of the liquid. Now the liquid that you've accrued will probably be around 800. So you're gonna take the pipette, switch it to 800. Now you're going to take the one with the pellet and the baby, open up your brand new microcentrifuge tube with a column in it. As you can see, it kind of like separates itself, this little thingamabobs, to the bin. I call this the bin. This is a trash bin. So I put it together. I open up our baby and pellet, pipette out as much of the baby you can without the pellet. Don't touch the pellet because that's the cell junk that we don't want. So right now in my pipette tip, I have the baby, which will go in the column. Column, baby, baby, column. All right, cool. So at this point, we can throw away the pipette, we can throw away our cell debris, which we don't want. The baby is safely secure in the column. Yeah. All right. So then this is kind of weird. This is kind of a weird shape. It's kind of like not really balanced. It's not, unless you want to waste one of your precious columns, which is unsightly. So what I do is I get just regular microcentrifuge tube, and I just fill it all the way to the top with water or whatever you're using. Saliva, blood, sand, actually not sand, glitter, whatever you want, okay? And you're going to carefully balance it on the centrifuge and then centrifuge it down very quickly. Oh no, oh my God. Oh my God. Okay. All right. It honestly doesn't really matter too much, but it says say 2 minutes. So centrifuge it for 2 minutes, and then as you can sort of see, oh shit, I dropped the baby. Oops, sorry. So the liquid was all up in here in the column, and now, as you can see, it dropped through the column to the bottom. So this is all the A1, the A2, and the N1 buffers that you used. Currently at this state, the DNA, the baby, is bound to this white little cotton column. That's where all the DNA is trapped at this point. So now you can pull it, separate it, DNA, junk. Throw the liquid away. empty. So then place the baby in the column back, and you're ready for a second centrifuge. All right, so then you're going to do 450 microliters of wash buffer. $450 of wash buffer of, let's see, DNA wash buffer. I feel like I'm missing something. I missed something. My bad, you guys. Sorry. So, so prior to the wash buffer, I don't normally do this. This is kind of like a weird protocol for me. But so prior to you putting the baby inside of the column, you actually coat the column with some column buffer. And the column buffer, you are supposed to add 100 microliters to the buffer, or to the column. You're supposed to coat that real nice and then just let it sit out for 2 minutes. And so once you coat it, it allows the DNA to bind to the actual spongy column, the little white little tip, rather than the walls of the column. I don't generally do this in my regular protocols. I've gotten very high yield from it, so it is an option that you use the column buffer but for the purposes of this course, your first run, you should use the column buffer. That's why we gave it to you. Sarah, going back, the baby, where is the baby? The baby is in the column. Yep, we threw away the trash, and now we put the column back with the baby. So then the next step is 750 microliters of the wash buffer. So we're gonna set that real quick. I'm so sorry, I've misled you. Okay, again, going back to the DNA wash buffer. Did I say 450? No, I put 750. 750, as you can see here. Maybe. 750 microliters of the DNA wash buffer. All right, we're gonna pipe at that. And we're going to add it to our column. E. And then we're gonna throw this away. So again, we're gonna take our balance full of water, poop, saliva, whatever you use, glitter, balance that really nicely. It's all balanced. And we're gonna spin it down. Okay. Oh, look here. So the water came down from it. Now the elution buffer has a little bit of ethanol in it. And when the ethanol goes through the binding column, what it does is it polarizes the DNA, so the DNA is still bound. It doesn't get like washed out. So what the ethanol is doing, it reinforces the DNA to be stuck on the column until you do an elution buffer or wash it with water. So the silicone here is actually positively charged right here, that little white thing. It's positively charged, and as we know, DNA is negatively charged. So right now, at the first step, the DNA was stuck here, and then after the elution buffer, it became polarized again, so it's extra stuck inside of the silicone little column here. So at this point, we're going to take them off, and we have the baby in here, and the wash in here. So we're gonna dump this out. So now we have room to spin down another time, not one more time, another time, because we want to get all of the liquid out. At this point, we do have a little bit of ethanol in here, maybe some like leftover elution buffer or whatever, and we should spin that all the way down so we have just pure dry DNA stuck in the column, ready to get eluted, finally. So we're gonna put this back inside the centrifuge. Now at this step, it's very important that we make sure that everything is dry. So I like to kill the horse over the head and bury it by centrifuging it for about 2 to 3 minutes. But I'm not gonna show you 2 or 3 minutes because I wanna go home and watch the Sharks game. So 2 or 3 minutes afterwards, centrifuging at 10,000 RPM, everything should be real dry. As you can see, there's like a little bit of liquid on the bottom, and it always comes out at the end. So you gotta make sure it's super dry. So at this point, it's very, very important to get a brand new column or brand new microcentrifuge tube, the ones that we sent you. They're just like the regular 1.5 mil tubes that we have. These are not the columns. So it's an empty, clean column, or empty, clean tube. And we're going to take the column with our baby, and we're gonna place it in here. Now, we're gonna talk about labeling real quick. You're gonna do this for one, but if you have like multiple samples, you gotta make sure to label always. So on the actual tube part, I'm going to draw a little smiley face for me. Tiny little smiley face. Yee. All right, so this is like pretty much the last step before you elute real DNA. So the DNA right now is stuck in the binding column. It's been reinforced with the wash buffer with ethanol, and it's just waiting to come out of the tube. So you're going to take 50 microliters. What is it? 50 microliters. So the pipette that I've been using only is 100 to 1,000. So this will not hold 50 microliters. We're gonna have to switch to a smaller pipette. And the smaller pipette, I believe, is 10 to like 200, I think. So we're gonna switch pipettes, we're gonna switch pipette tips. Actually, it's 10 to 100, my bad, it's 10 to 100. 10 to 100 pipettes, pipette tips. So we're going to crank it down to 50, is what we need. Okay, put some tips here, which don't fit. One moment. Don't fit
There's
either.
the pink one here.
Oh, pink one. Thanks, Jill.
No? Oh my gosh.
Oh no!
No. Crap. Give me 1 second.
Oh, actually, let's do this. I'm gonna take my other pipette. Obviously, the Ferrari of all pipettes, the Rainin. Same thing. It's okay. It's fine, you guys. And I'm going to crank it to 50. And use some different tips. Okay? So we have 50 microliters that we want to get the elution buffer. So carefully at this step, it's very important to put the elution buffer straight onto the baby on the white portion. Make sure the tip doesn't go all the way through because you can actually puncture the silicone. So what I do is I kind of carefully pipette up the elution buffer, put it straight onto the cotton, but not actually touch the cotton. It's really hard to visualize here because it's very, very small macro things that I'm working with right now. But the reason why I'm doing this is because I don't want the liquid drop to be like on top or like on the top of the column, because at this point the elution buffer binds to the DNA in the column and then it pretty much depolarizes it from the column and it starts eluting through the bottom. So now, yeah, try it out. You get more yield if you put it straight on the column versus like you just throw it on top, which is something you don't want to do. So yeah, let's take a stab. You're gonna open it up, pipette some of the elution buffer, and the best I'm going to show you, pipette, insert. Couldn't see that at all, but it's cool. It's right, I pipetted right on top. Close that up, throw that away, close up your elution buffer, and at this point we're going to wait. We wait because we want to ensure that all the elution buffer is bound to the DNA so that we can have a really good recovery rate. So once that is done, once you leave it for about 5 minutes, then we're going to place it back in the centrifuge. Again, it's balanced with that full-blown weight that we put in. Opposite sides. And you see like the top is kind of like a little like out. You can kind of like tuck it in from these centrifuge tubes, so you can see it's like a little tucked in right here. Because if you like leave it out like this, it might break off during the spinning. So you're just gonna tuck it in real nice. You're there, and then you're going to centrifuge it out. Again, for the sake of this tutorial, I'm not going to centrifuge it for 5 minutes. All right. Now, that is the prize! The beautiful purified baby. It's right there. That's so cute. The baby. Yes, indeed. If you prefer puppies or kittens, I totally understand. I'm also a millennial as well. But the baby is right here. So now, at this point, that is your last step. The column gets tossed away. This has no baby in it anymore because we depolarize the DNA from the column to be free-flowing because it dissolves the DNA, kind of like mingles with the water and breaks down with the water. So it's been eluted. So currently we have right here is pure water and DNA. So we're going to throw this away finally, snap it shut, and there we have it. We have a purified DNA inside of microcentrifuge tube. You can do a myriad of things with this. All right. Okay, I'm gonna come over here. Thanks, Jill, for holding my camera. All right, so I took you through the steps of a mini prep, and it was quite long. I'm not gonna lie. What time is it? Oh my god, it's been an hour since I've been talking. So yeah, it's a very long process. It's a very tedious process. I've probably done like hundreds and thousands of these while I've been working. It's an essential staple to genetic modification because you can't just literally take an organism and just be like, ha, like DNA, exit. you actually have to go through lots of steps to purify the DNA. So once you get the purified DNA, you're going to, number one, you're going to run a gel electrophoresis. Now, gel electrophoresis, I believe, is in week 3 for you guys. In gel electrophoresis, you actually need to build your own gel phoresis box. And we have all the steps for that in the Google Doc, or in the Google Classroom. So take your time building it. It takes a little bit of like just like wire engineering, and then test out some of the casts and the little combs that we have provided for you. Practice making an agarose gel. Make your buffer, your 1xTAE buffer. So I will show you next week how to build the box really quickly, run the DNA on the gel, and visually show you the DNA that you've isolated. So at this point, right here, what you can do immediately from this is, number one, you can gel electrophoresis it. Number two, you can just store it inside of a freezer, because DNA can be frozen for a long time. It can be frozen up to 6 months before it starts degrading. So you don't want to leave this out on the lab bench too much. It will degrade much faster. But if you put it in the freezer, freeze it, it'll stay way better for much longer, and you'll be fine with that. And you can defrost this anytime and run a gel for electrophoresis or do genetic engineering or whatever you please. So yeah, that kind of concludes the mini prep.
Side note, T. Williams was asking about his ligase and all of the stuff basically that's in the stuff you need to keep frozen, the negative 20 degrees Celsius. Yes, there's a tiny amount. You say, like, it doesn't even look like a full drop. It's like a half a drop or a quarter of a drop, and that's fine. If you don't see anything, let it thaw, put it in the centrifuge, of course, balancing it, and then see if you get anything at the bottom. If you see absolutely nothing in the tube, then go ahead and email us.
Yeah. Thanks. Yeah, what scientists usually do is we take the tube and it looks like there's nothing there, like 100 microliters or 50 microliters. It's very hard to visualize, especially if it's stuck on the side of the tube. So after you take it out of the freezer, what you can do is you can hold it, or you can put it in your armpit, or put it in your pocket. Just make sure you don't forget it, and it'll thaw out, and then you'll see like just splatterings everywhere. What I do is I flick it again, flick it, just flick it once, just and everything should go towards the bottom, which is a nice way of doing it very quickly without like centrifuging it and balancing it. So it's a quick, dirty method, works wonders. Yes. Let's see, anything else I need to mention? Any questions thus far? I'm gonna take a bunch of questions right now. I'm sorry I blew through it kind of quickly. Again, I really want to see the Sharks game. Go Sharks. T. Williams, we're not going to go over gel electrophoresis today. I'm going to go over it next week. Yes, there is a series of steps before you load it into the actual gel. Again, I'll go over all of that next week. I'm just going to go over incubating your bacteria and also mini prepping just for today. For your nuance, can you cut DNA segments out of the gel and use them? Yes, I will definitely go over that, but you can essentially isolate a portion of the gene or like something of interest with a particular size, and you can actually physically cut that out of a gel, mini prep that and purify it again, and you can use that small fragment of like a gene that you're trying to find and purify that again and then reuse it for genetic engineering to put inside of a different organism. I've done that lots of times. It's worked wonderfully. Yeah, it's just a really weird step that you can take. But I'll go over all of that stuff next week. I'm just teaching you guys how to mini prep. Okay, Julie Miller. What's up?
Another side note, your parafilm is in a little baggie in one of the padded envelopes. I know we kind of hit it. We probably should have put directions in there, but that's where it is.
Yes, parafilm. And very, very important. So yeah, Julie Miller asked, how do the buffers work? I'm gonna go over it very, very quickly. It's a good idea on how the buffers work. They're not just labeled A's and N's and 1s and 2s and 3s. Again, this is something to reinforce your learning. This is a good method to put on your resume. Doing mini preps, doing DNA purification. It's just such a large thing practiced in industry. Again, it's essential for genetic engineering. You can't genetic engineer without pure DNA, or otherwise it'll completely screw up your results. So this is a very important thing that I'm talking about. But I will quickly go over some of the buffers. So again, we have the column. It's completely neutral in charge except for the tiny little white part that is positively charged and DNA is negatively charged. And so once you put the DNA inside of the column, you elute it out. Or I'm sorry, before all of the column stuff, we have buffer A1, we have buffer A2, we have buffer N1. So buffer A1 is essentially something to suspend. It's a very neutral buffer. It's to suspend your DNA, to suspend your cells in a very nice little warm bath to kind of loosen up the lipid cell walls. And that is the A1, and it's just a really nice like pHer just to kind of get everything mixed up pretty well. So that's A1. A2 is like it's a harsher emulsifier. So this is generally made with I can't really think of the chemical at the top of my head, but it's pretty much a lysate. And what it does, it lyses the cell and starts the cell separation from the DNA. So all the cell junk we don't want, for example, all the organelles, the Golgi apparatus, the lysosomes, the mitochondria, the cell wall, the cytoplasm, just like all the stuff that we don't want versus the DNA that we do want gets separated at this moment when you immediately add the A2, which you can see that because it forms that really cute little booger. And yeah, it's just like kind of jellyish, and you could totally tell that the DNA is like very, very like thick and viscous. And at this point, everything is kind of getting like jumbled up and being lysed, like all the cells are exploding. And then the N1 is pretty much the ender of all worlds, the destroyer, and it will completely separate the DNA from the cell junk. So even if you add N1 and you leave it on the bench, and if you leave it for maybe 10 or 20 minutes, you can actually see a full separation of DNA from the cell junk. So the cell junk will actually float to the top layer, and it looks like kind of like, when you're like cooking like bone beef broth, you see like that fatty layer just like separate from the liquid, and it's just all the fats and proteins that just kind of skim at the top. So the N1 does a really great job of doing that, and the DNA is located within the liquid. So then you take that liquid, baby, puppy, kitten, whatever you want, putting it in this column, eluting it, throwing it away, taking the column and the baby, washing it with the DNA wash buffer that has the ethanol. And ethanol, again, is very polar, so it will not allow the DNA to be eluted once you throw it in the centrifuge, and so it'll be very, very stuck to the column, which reinforces the DNA to be stuck to the column. And then at that point, we take the elution buffer, and then we elute it with the elution buffer, which contains mostly water, a few salts and a few buffers, but it's mostly water. And then you depolarize the DNA to come and elute out from the column. And that's what the pathway of the DNA is doing in the column. Yeah. All right. I'm trying, Matt Barnes. Jesus, I want to watch the Sharks game. It's, by the way, it's Game 7 for the Sharks and the Avalanche. I can't believe we've come this far. Goddamn. Should have ended a while ago. Ugh. Buzzwordy09. How do you test the DNA? Nano drop? Yeah. Unfortunately, we, even the ODIN couldn't afford a nano drop. I think a nano drop is like $5,000, anywhere from $5,000 to $10,000. So a nano drop is a really nice instrument that you can drop 1 or 2 microliters of your purified DNA, and you can actually quantify how much DNA you have. So, yeah, it's a really great tool, especially if you're doing genetic engineering. You want to know how much DNA and how much concentrated that one little tiny drop is. So for example, if it's too little of DNA, you can actually use a nanodrop, quantify it, and then say like, okay, it's too little, so maybe I'll elute it next time in like 20 microliters versus like 50 microliters. Or if it's too much DNA, you can elute it in like 100 microliters versus 50. It all depends on the nanodrop. Now, since we don't have a nanodrop that we're going to be working with in the scope of this class, it's not super important. However, when you are doing some like much more advanced elution, or even like telling that you have DNA, it's really important. So again, for the scope of this class, don't need a nanodrop unless you want to sink $5,000 into it. You can also quantify DNA through the means of gel electrophoresis. So again, when I go over gel electrophoresis next week, you can load the DNA into the gel, and you can run the gel out, and you can actually check the intensity of the bands, like the fluorescence of the loading dye and the DNA bound to it. And you can actually indirectly measure how much DNA you've gotten, like a lot or a little. You obviously can't numerically like say, oh, it's 79 or like 150. But you can tell that you have gotten DNA or you haven't through gel electrophoresis. And again, I'll show you that next week. Okay, so let's see. T. Williams says, does the last buffer also unwind the DNA like from the histones? Nope. I believe at that point it's single-stranded. I believe so, yes. Buzzwordy09 says, I thought they need the numbers to take it from sequencing. Yeah, so sequencing companies, these are great questions, by the way. Sequencing companies, they generally ask like the questionnaire before you submit your sample in. They're like, okay, like how many base pairs is it? Like what's the concentration of it? And you would use a nanodrop to measure the quantity and then to report that. But generally, like, since we don't have a nanodrop here, we send it out for sequencing, and we send them a lot. So we'll send them probably like 50 microliters, what we eluted today. We'll send the whole thing out because number one, we don't need it. We just need to verify that it is correct DNA and that you can synthesize from it. So it's not super important for us. It might be important for the future when you do want to send it out for sequencing to quantify it, but it's not that important because if you send it into them, they'll actually nanodrop it themselves. That's the first thing they do. They take 1 microliter of it, they'll nanodrop it, and they'll internally themselves quantify how much DNA you eluted. But as long as you're sending in like 50 to 100 microliters of your product, your purified product, it shouldn't really matter that much. Yeah. All right, without any further questions, stay tuned for next week. I will see you guys on Tuesday of next week. And we're going to be going over gel electrophoresis. So make sure before our class to build it very quickly, to run an agarose gel to create TAE buffer. So you can kind of be familiar with like what is going on, and then in the tutorial I'll teach you how to load properly everything, anywhere from like DNA loading dye to all the way to like the ladder. So we'll be covering that all next week. It'll be at the regular scheduled time. Again, I'm sorry that the ODIN had an emergency yesterday, that we had to run and do something real quick. So sorry again, but thanks for tuning in. I hope to see you soon. And make sure you reach out to me on social media, on Facebook, Instagram. I am all over the ODIN page. If you want to message me through the ODIN page, like, feel free. Again, my email is at esther@the-odin.com. Cool. I can't wait to see the rest of you. Bye.